Hello and welcome to the final IBMS podcast of the year. In this special festive episode, we're joined by quality control scientist and bake-off star Yan Tsou to talk about cell lines and brining Christmas turkeys, after which former biomedical scientist in haematology, Reverend Christopher J Lee joins for LabLife. Hope you enjoy and see you again in 2022.
Two IBMS members have been recognised for their outstanding record of continuous professional development at the Science Council CPD Awards 2021. Congratulations to IBMS fellow Mark Sioni, who won the CPD Award in the Chartered Scientist CSI category, and Victoria Mosey, who was commended in the same category. The latest edition of our Superlab comic is now available as a free downloadable digital copy. The comic is for Key Stage 2 children and features all sorts of fun biomedical science-related activities. Download your free copy via our website now. Earlier this month, our members took part in events up and down the country to celebrate National Pathology Week 2021, which took place from the 1st to the 7th of November. You can take a look at the highlights and our online news roundup now.
Hello and welcome to another IBMS podcast. Today we are joined by Yan Tsou, who works in quality control at the Francis Crick Institute's Cell Services team, but many of you will recognise her from the Great British Bakeoff. Yan, welcome to the IBMS pod.
Oh, hi.
Hello. I'm going to hand you over to my colleague Jordan, who's going to get things going today. Jordan, over to you.
Yes, so thanks for joining us, Yan. Welcome to the pod. And we've just heard from Rob in the intro that you work in the quality control team in the Francis Crick's Cell Services department. So could you kick us off by just telling us a little bit more about what is the role you do there and why is it so important?
So we're in a little team as a side offshoot of Cell Services. So we're able to do the work of Cell Services, and the main function of Cell Services is to provide cells to the research scientists to do their experiments on, in a nutshell. So, but also what happens is that when research scientists have manipulated the cell lines to what they want them to be, so like they take some genes away, add some genes in, or whatever they do to it, manipulation, they then rebank them with us, so that when we're a sort of like a storage facility for this facility as well. So what happens is when they return them back, we then put them through a barrage of tests, so to make sure that they're nice, clean and healthy, and so no mycoplasma, which can affect the cell line's growth and also genome sequence. And also we prove they are what they say they are, because, and I tell you this to students that come in that learn about cell work, is that cell lines pretty much look similar under the microscope. You can't actually tell that much difference from one to the other. The growth rates are different. So like human cell lines grow a little bit slower than animal cell lines. Say like mouse grows a lot faster than human cell lines. But other than that, they all just look like little blobs under a microscope. So what we do is we use PCR to not only test for mycoplasma, so we test for mycoplasma using PCR, and also use PCR to species identify. So we can species identify between different animals and human cell lines. And then we then go further testing, because if it's a human cell line, we then go further testing to identify what type of human it is. Because like there's different types of human cell lines. So I don't think you've heard of things like HeLa, which is your HeLa, HFFs, human fetal foreskin, 293. So that these are from different humans. They originate from different humans. So therefore, we use PCR, a technique called STR, single tandem repeat testing, to identify the human that has given that cell line. And we have, in the past, so to give you like a few horror stories, is that they can either submit and think it's their cell line and have been working for it for several weeks or whatever, and then it comes to us, we test it. They are either, and then it's, as long as it's clean from mycoplasma testing, we then test for species, and then we find maybe mouse cell line in with a human cell line. That can happen because if you're sharing, as here, the research teams share equipment and stuff, and accidents happen, and you can accidentally even dropping one single mouse cell into a human flask, human cell line flask, it can outgrow a human. So like I said, accidents happen because equipment is shared and the rest, but more or less, usually 99% of the time, it's exactly what the researcher says it is. And then what we also do is, if their cell line is coming from outside, so not come from us, it's coming from an outside source, the research scientists are very good. They submit their cell lines for testing for mycoplasma. So we have a three-pronged attack. We have PCR testing and fluorescence and agar testing. So we don't rely just on the one technique, because different species of mycoplasma can present over one technique over another.
Okay, so mycoplasma is the most frequent contamination you see within these cell lines that are being used across the entire institute.
Yeah, but it's the contamination that can infect the cell line without you really realising. If it's contaminated with like bacteria or spore or fungi, you spot it straight away because it'll outgrow, and also like the smell is atrocious. But what it is that those contaminations will kill the cell line. Mycoplasma is more like it's a bit not. You don't realise it's happening, and you might realise because it's sort of like it slows down the growth of the cell line. The cell line might not grow as well, so people can spot it but not have actual proof that it's there. And we have like technique to test it.
Okay, it goes under the surface a bit more. It's a bit more invasive contamination.
What's important about that is because there's different types. It's like so mycoplasma can be slow growing or fast growing. And slow growing, it can be in with the cell line for ages and ages. And the whole point of detecting it and eradicating it, because you can eradicate it, is that while it's there for any length of time, it can change the sequence of the cell line. So if the sequence of the cell line is important to the researcher, they do not want mycoplasma to be in there infecting.
Have you ever come across an instance where it's actually infected one of the researchers' cell lines undetected and it's affected their research project, and how?
We test it, and it's quite funny because we test it and we don't really know it's our job to tell them. Yeah. We don't really know the outcome, but from my experience and now my experience of our team, because we're a unit, is that sometimes it's quite difficult because the different species because there's various, I can't remember how many different species there are. It's a bit like mycobacterium is like different species. They have slow growers and fast growers. The slower it is, the harder it is to detect, because you can only detect over a certain concentration. We use the three-pronged attack. We use PCR, fluorescence, and agar, because they're very different techniques. And the PCR can pick it up to an extent, up to a certain concentration. Fluorescence can pick it up to a certain concentration. And agar, because you leave it for a long period of time, can pick it up at very low concentrations. But with those three different techniques, there's like pros and cons. PCR, it takes like a few hours. Fluorescence and agar, fluorescence takes 3 days and agar takes like 2 to 3 weeks. 4 weeks now, extended that period. So
you'll pick the best one depending on the needs of that moment in the lab.
I mean, what we tend to do is that we, as a strict rule, we do fluorescence and agar, no problem, because it's very cheap and easy to set up. But PCR, expensive because it's fast. And actually, it picks up quite a low concentration. But it's within that, because it's expensive, we have to limit we don't test absolutely everything. We have to limit it. All budget related, isn't it? I mean, we all know what to do.
As it is most of the time in science.
Most of the time, yeah. It would be great if we could use PCR on everything, but that's simply not possible.
Just a couple more things I wanted to cover before pass you over to Rob to talk about baking. What was your journey into this role? What did you study originally, and what's your kind of background in biomed?
So originally I was at the University College London, which is just across the road from here. I did medicinal chemistry because I thought, that's really exciting. It's to design new and novel drugs for therapy and all the rest. Turns out I'm not great at chemistry. I'm okay. I mean, I got a degree out of it, quite a long time ago. So we're talking about, I graduated in 93, got my degree. I just sort of like drifted from like one. So I started off like just applying for jobs just to get experience, because in those days, experience, you can't get a job, and you can't get the job without experience. So it was like a double-edged sword. And what I did was I volunteered at a cancer research place in Hammersmith at Charing Cross Hospital, and then just learned cell work there. And then, funnily enough, this isn't the first time I've worked here. Worked for this department. In its first incarnation, it was at South Mims, Cancer Research UK. So I worked for this department in 96 to 2000. And then I went away, learned PCR, because at that point in time, they didn't have PCR machines or anything like that. Went away, learned how to do PCR with blood service, became a biomedical scientist, because it was then we had to all be registered. We had to be state registered, and then did a stint at the TB lab at Public Health England, which you also need to be state registered to work at. And then I came full circle about 4 years ago, back to here. I started here just before I started Bakeoff, which was nice. They actually, my boss, who I've known for like over 20 years, was able to give me more leeway than I think I would have done if I was still working in a government role.
And has science always been something you've been fascinated by since you were like a kid, or was it something you kind of it came a bit later on?
Yeah, I love it. Do you know what? I love all aspects of it, but I really love being a microbiologist, biomedical scientist in bacteriology, microbiology, all fascinates me. And I got into not a Twitter argument, a Twitter discussion on why, like yesterday, on why COVID isn't like chickenpox. Because they were saying, if you get infected, you can get it's just like chickenpox, you become immune forever. And I said, well, actually, chickenpox, you don't become immune, it stays in your body forever. And then if you become a little bit unwell, like a little bit immunosuppressed, it comes back as shingles, and then you can become you can infect other people with chickenpox. You won't, infect other people who's had chickenpox, but you infect people that don't. Yeah. And I sort of not got any, but because I'm not a trained virologist, but it fascinates me the whole. Yeah, because viruses are fascinating, and if they weren't so deadly, it'd be like their mechanism and all the rest. So everything like that interests me, how bacteria works, how mycoplasma works. I just, it's always learning.
Well, one more from me before I hand you over to Rob. It's just a little bit more about the research papers you were doing, because in 1 year you were named as an author in 5 published papers, including one in the Lancet, and you published a recipe in the British Bakeoff cookbook all in 1 year. So well done. And what was the research you were involved in? Can you tell us a little bit about that before I hand you over to Rob?
You know what? That was with the first incarnation of Public Health England. So I worked for the one before that, because they renamed themselves Public Health England, but it's actually the Health Protection Agency in the TB unit at Whitechapel. And such a great project. That was a great project. And my boss then, Professor Drobneski, he hired us to oversee a project called Predict. It was for immunosuppressed patients that could become TB, if they had latent TB, what would it take for the active TB to pronounce itself, to manifest?
Oh, right.
And so that is basically a Pandora's box waiting to be opened. That's what he described it as, because the entire population, latent TB doesn't manifest unless you're immunosuppressed, or it may never manifest. And so there's a test to test for latent TB, but if you start testing, then where do you stop? So they started testing the communities where it would be most prevalent that they would have latent TB. So we were testing so the amount of information that came from just taking blood samples from these groups, individual groups, mostly around like East London, different ethnic. So it was ethnically diverse as well. So like just and also where else was interesting? The homeless community, they were taking blood, because what it is that if they're in a home, like in a hostel, it's a crowded situation. They've been immunosuppressed because they're out in the open. So it's a lot of factors to take in, which is why there were so many papers, because there was a lot of different approaches to this sort of research. All the samples were taken over a period of 2 years, and I was doing 30 samples a day for over 2 years. So we had a lot of data, and from that data, they were able to approach the different aspects of latent TB and how to detect it. And that's why there were 5 papers. 5 papers plus possibly more, I don't know. And then I was able to put them on a CV.
What's the significance of identifying it on a large scale?
That was exactly it. That was exactly the question. Because what it is that from this then research, I suspect that what they were trying to get at is, do we then go large scale, and who would bankroll it? It's again, we come full circle back to the money. So it's and that's why my boss at the time said it's a Pandora's box, because if you don't know, then you don't have to act on it. But once you do know, you then, with morality dictates that you should. Like at the same time, it was fun in the sense it was really interesting. You know, like everything interests me. That was like going down that rabbit hole, and I mean, joking aside, it's a really, from a sociological point of view, and talking about housing. So then you have to go into like sociological, like housing, because even though there shouldn't be, there are pockets of the community where people are overcrowded in their homes. And as you well know, is that if you've got an infectious person, if you're living in a mansion and you're infectious, you're unlikely to have communicated that disease to somebody else. It would be unlikely. There's no gold standard of sizing of housing and rooms and all the rest of the overcrowd to affect one on the other.
We're going to talk now a bit more about baking and science. So I'll hand you over to Rob.
Yes, thank you very much, Jordan. I've got the good job of going from latent TB, morality and socio-economics straight into the Great British Bakeoff, which is a very natural transition.
So probably fast forward it all the way to like the middle where we go, what about the baking?
well, let's jump back into it. I think it was 2017 when you were on the show, Yan.
Yeah.
Tell us about how you got onto that show in the first place, and whether science played a role in that.
Science didn't play a role in why I wanted to go on the show. And I'll tell you a funny story. What it was is the season before, my wife and I was watching The Extra Slice, and they were giving out those cakes, you know, where they look like you. And I turned to my wife and said, I want one of those. And that's where it grew. And then what it was is that year, it was the year before I saw the cake. And then that year that I could have applied, I didn't, because I was studying for my master's in biomedical sciences. And I said, and it's really strange how things say, because I say a lot of things, but they don't come true. But I said to her at the time, I said, I won't apply until I've got my master's. And then I got my master's, and then Mary said, "Oh, are you going to apply now?" I said, "Yeah, I guess so. But if I will apply this one time. It's season 8. Apply this one time, and if I don't get on, I'm not going to apply again." And then what it was, and I think I told that story to the people that were like at the audition, and one of them said, "Well, we better get you on then, hadn't we?"
If we don't do it now, never.
I'm not going to do it again. Because it's season 8 is in Chinese culture, number 8 is lucky. So I took it as like, but also the previous year, because I was still working at Public Health England, I was doing all the technical bakes that they were doing and then bringing in the next day for work. So I was baking the technical bakes, obviously not now that I know that technical, but I use the actual real instructions because they publish it afterwards. But I was baking it just as proof that I can do the technical bakes. Let's see what I can do for real. But yeah, it was that's how I got on.
How did you find juggling? Because A lot of the people have got full-time jobs. Some people don't. I know that you were working throughout. How was that? That must have been very stressful and tiring.
Oh, it almost killed me. It was a good job I was that age, because I don't think I could do it now. These days, I could not do it now. It almost killed me. It was that. So what it was is that you have to practice, and you sort of know what you're baking. So they gave you a place, they give you the brief and all the rest. So and then you're practicing and practicing and practicing. Once filming starts, and this was before we was in a bubble, so I couldn't stay there permanently and practice there, which I don't think that would have been easy either, personally. But what it was is I was coming to work 9 to 5, getting home, bombing down my dinner, staying in the kitchen, which, by the way, was like a tiny. It's been refurbished since. It's been taken 2 years to, like, we've had a renovation since, but like it was tiny kitchen in a 2-bedroomed Victorian terrace, a really old kitchen, freezing, and my oven was lopsided. So in order to make my bakes level, I had to put a spirit level in there to make sure, and prop it up with a wooden spoon. So this is all filmed, and actually to shut the oven door, I had to stick a bulldog clip to keep it shut. That was how I started. And then what it was, I would bake and bake and bake until like 1 or 2 or 3 in the morning, go to bed for a few hours, wake up, go to work. So in between, that was what I was practicing. And that was, oh my God, that was so knackering.
And you made it, were you knocked out with 3 rounds to go, is that right?
Yes.
Have I got that right? And were you glad when you got knocked out?
Lay down. Sorry?
Were you glad when you got knocked out? Were you thinking, my God, I can finally have a bit of a rest?
You know what?
Or did you feel really disappointed?
No, it was literally, by that point, I was like, thank God. And I think, because you're mic'd up the entire time. And at one point I was walking across the quad, like the grassy bit. And I said, right, you lot have to stop it now, because my job, I love my job. This is a fantastic gig. As in, like, my bosses are really supportive. You don't really get how good it is unless you've been to somewhere really. I've been to some. They're okay. I'm not going to diss anybody else, but this job is. I work with my best friend, so I was saying to them, like, in the mic, because I can't see me. I said, right, this is ridiculous now. If I lose my job, what does this get me? This gets me nothing. And so, like, I was just talking to myself and whoever was listening at the other end of the mic. I don't want to lose it, because I've worked very hard to get where I am. 5 papers is no mean feat, you know.
Yeah.
1 year, because there was and that's not included, you know, like, that wasn't including the one that we had in Nature to do with sourdough, so, which I'm most proud of. I didn't even write that one. So yeah, it was tough, but I was relieved. Yeah. Because I think, you know, like, if I won it, so what? What does that really, you know? I know there's people who've been successful, but that's not what I wanted out of it. My whole aim was to get that cake of an extra slice. And I got that.
Hey. And you were quite scientific in your approach on the show, Yan. Let me talk. I've got one of the titles of one of your bakes here in front of me. It's mango salmon roe cake made with liquefied agar gel. So how much did the scientific approach actually affect your style of baking in the show and in everyday life? Well,
so that one was the banana ramen. So it had to be banana cake, even though it's not my favourite cake. It wasn't my best cake, but it just for the pun. I wanted, because I knew it wanted to be. Do you know what? It wasn't even the ramen that started me off. It's the liquefied agar.
Yeah.
The mango agar balls. It's because I was working here and making agar every day to make, you know, like we run those PCR gels.
Yeah.
The classic PCR. So we put under UV and see the bands. That, because I knew that recipe off by heart, I knew that's what I wanted in the cake. And from that, I built around, what can I do? What can I do? Because it was a illusion cake. That was illusion cake. What can I do? It was either that or sushi. But a bowl of noodles looked better, and actually was achievable in the time that they told us. So that was when I was still sort of like toying with the idea. And then I thought, all right, so it's going to be ramen. No, and I think banana ramen had just come back and forth. So I went, banana ramen. Okay, it's going to be banana cake. then just carried on from there. But
Nice.
it stemmed from that one, the agar that I make every day here. Obviously, I didn't use this agar. I use food grade agar.
Yeah.
But the recipe's still the same because it's a 1 in 100 with water. So I knew what the recipe. I know what the recipe is, and then had to experiment with the mango because if the mango was too sour, it didn't set. So then I played around with the ratios.
Brilliant. Well, that seems like a really nice note to take you on to a little quiz we've devised for you here, Yan, which is called "What's on Your Plate?" And it's Christmas themed. So on Christmas Day, do you have a starter?
This year I am because I saw online this guy. He made like it is a brown butter, and it's a really simple but tasty. So it's not really a starter; it's a side dish. And I really want to do sourdough on that on the morning of Christmas because it just makes the house smell so lovely. So yeah, it's going to be brown butter and sourdough.
Nice. And question number 2: what's for the main course, Yan?
So we've got 10 coming round, but actually it doesn't matter about number because I still always make the same meats. So it's going to be a rolled turkey breast, rib of beef, and a belly of pork.
Oh, delicious.
All the trimmings made from scratch. Well, apart from the parsnips, made from scratch.
And what about afters? Anything following that main?
Yeah, always. At least 5 desserts. There's going to be 5.
5 desserts.
There's always a trifle because Dad loves a trifle. There's going to be a roulade, a meringue roulade, Christmassy type thing. Mum loves profiterole, so I usually do like a crackling Christmas flavour. But again, these are made over, so I don't make them all on the same day because that would absolutely kill me. So I would make the profiteroles the day before and freeze the week before and freeze them. So that's profiteroles. What else? Chocolate cake for the little ones because they only like chocolate cake, and something else, a mystery one. So it's at least 5 because everybody likes something different, and I want to make everybody happy, which is the whole point of Christmas, right? It's not about the presents. No longer. Do you know what? I'll tell you something. It's no longer about the presents. Because since we had our house refurbished, we've got a lot less storage space, and we discovered that we didn't need any of the rubbish that was in our cupboards or wardrobes that we got rid of. Don't need it.
Nice.
Good food, good company.
Well, I've got a few more quick questions for you, and these are a bit more science-based, Yan. So we'll hopefully be able to help out some of our listeners at home.
Or not.
How do you stop a turkey drying out?
Do you know what? I would say brine it.
Yeah.
As long as there's no health issues with people overdosing on salt, I would always brine the turkey. Because that's what we do. We brine the chicken.
Nice, good tip.
So get the brine correct because you don't want over salty. So use a recipe and weigh, because that's weights and measures as well. Weigh your turkey and do the brine concentration correctly.
And roast potatoes, how do you get them to crisp up, Yan?
So use the right type of potatoes. So a dry weight content. So we're talking about. I find people swear by King Edwards, but I prefer Maris Pipers. So and if you're listening in America, then I think Yukon Gold is the one that you go for. So it's a higher dry matter because you don't want anything that's too waxy because it will be soggy potatoes unless you like that sort of thing. And then, so do you know what I found is that roast potatoes are essentially chips, but because they're crisp on the outside, fluffy on the inside, and so you really hot pan, hot fat, and cook. They always take longer to cook than you realise.
Yeah.
Yeah, because if you think, it's going to take, or you turn up the oven at the very end and put in the pan and heat it takes, if you think it takes 15 minutes, it's going to take 30 because we always, no matter what, and after 20 odd years of cooking Christmas dinner, worked out that we've always been waiting for the roast potatoes. So now I do that. So another secret is if you do them in advance, finish them off in the air fryer.
Oh, nice.
If you've got one, obviously. But these days, air fryers like my godsend.
So next up, Yan, just two more of these science baking questions. How do you stop your Christmas cake from getting burnt? Whenever I make it, the outside always burns and the middle is raw.
So, do you know what? I've never made a Christmas cake, but from what I can gather, it depends on the size as well. If you've got like a standard cake tin, what's about 8-inch cake tin, it's low and slow because it's a denser cake, and actually there's high liquid content as well. So low and slow. So if you're going at 170, take it down to 150 and it's longer. And then what it is also is that, so Val taught some of the things that she taught is incredibly useful. She says, listen to the cake. So if it's like going bubble, 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 it's not yet done. If it's going plop, 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 you can actually switch off the oven, cover it, and it'll cook in its residual heat, and it'll be nice and moist in the middle. That's the same for like it. Not so good for if you're making brownies because you never you can. If you do that, you can overcook the brownie and become a dry mess. But like Christmas cake and also a probe, cooking probe.
Temperature probe, yeah.
Yeah, I think you've got I can't remember what the temperature is. So my friend James, he makes Christmas cakes every year, and that's what he uses. He uses a probe. And so what you do, you go into the densest part, middle, it will tell you what temperature it's reached.
Nice.
Like 100 or 90 something. I can't remember. But yeah, he's got it on his if anybody wants to follow him, he's Cluck Muck Cook and he's on Instagram. So he has like a tutorial on his one. That I
Brilliant. And finally, why don't people like sprouts, Yan? I don't know if that's scientific or not, but why don't they, and do you like them?
What it is depends. So like people can say they don't like rice, but that's because they have the wrong type of rice. If they don't like rice, it might be because it's been cooked the wrong way for them, or they've had like bad rice. It's like relationships. You're not into relationships. You've had bad relationships all your life. So yeah, sprouts is because you're cooking them till they're like they've changed colour and they smell like farts.
Yeah, good stuff.
So what I'm saying is al dente, just cooked in the middle. You toss it with a little bit of butter. Delicious. So like in Far East, a really similar dish. There's it's Brussels tops. So it's exactly the same plant, but what it is, they then slice it up and then just stir fry it with a bit of garlic.
Oh, nice.
It's delicious. Oh my God, it's so good. Stir fry it. Chop it up and stir fry it with a bit of garlic instead.
Nice. Break some rules this Christmas.
Yeah, exactly. But also what it is that people eat sprouts because it's tradition, whether it is yucky or not. So, but we buy the Brussels sprout tops because it tastes so much nicer.
Fantastic. Well, I'm going to pass you over to Jordan now for the quick fire round. Jordan?
Thank you, Rob. Yeah, so we usually end with this round, just a quick round of rapid questions. So, okay, so are you ready for the first question? So, if I had to eat one meal for the rest of my life, what would it be? What would it be for you?
Would it be for me?
Yeah.
You know what? Roast dinner.
Roast dinner.
I love roast dinner. So versatile. Yeah.
Good choice.
As long as you have to cook it.
So the next question is, what piece of advice would you give to someone starting out in baking?
Learn how to make bread. Make bread and everything else will follow. If you can master bread, then you can master all of it. I love that's personal choice because I love making bread. But what it is that if you make bread by hand from scratch, no machines, don't use machines, use your hands, feel it. That's what I would advise. And then from there, you can make your bread, sandwiches, pizza, calzone, because it's versatile. Just the one dough can make all these things. Amazing.
That's really good to know, actually. And then the same question, but this time, one piece of advice you'd give to someone starting out in biomedical science.
I don't know, because I was lucky, and I'll tell you the honest truth, I was lucky being in the right place at the right time, because where I was when I was, we had to be state registered, I had a very supportive network. So this is a good piece of advice. If you think that it's not for you after your first job at any lab, try a different lab. it's like what was I saying before about you having the wrong, if you've had the wrong rice or wrong Brussels sprouts, it's because you it's been done, like it's not quite right for you at that moment in time. Don't be disheartened with any job if it's like your first job in biomedical sciences, because there's the field is vast and wide, and you just have to find your nirvana.
Absolutely. And a few more on these questions then. The favourite dish I baked on Bake Off was?
Do you know what? I really enjoyed making that pie, that showstopper pie, because I was just having the time of my life. I loved it. I loved doing that one. Oh, and the caramel showstopper. Do you know what? I loved... So this is the thing you've got to know about Bake Off. You've got to write the recipe. I loved writing the recipes for the signatures, but they were a little bit more boring. Loved doing the actual showstoppers whilst I was in the tent, but I hated writing them up because it was so faffy. It was like, you've got to remember this, got to remember that. I knew how to do it, but I had to write it out so that they knew how I did it.
So a final couple Christmassy themed ones. My favourite Christmas movie is...
Love Actually. I know it's not everybody's cup of tea, but I just love watching it. It's not Christmas unless I watch Love Actually.
The final sentence is, on Boxing Day, I always...
have a sandwich. Boxing Day sandwich.
Turkey sandwich.
Yeah, you know what, right? So it's the same as in the tent, same as out in real life, is I taste, if I'm doing a big meal for other people, I taste everything. I've made sure, and if you saw the offcuts during the show, I was constantly putting something in my mouth, just eating and tasting and making sure it tasted right. Is I do that on Christmas Day. And so by Christmas lunch or Christmas dinner, I'm not hungry anymore because I've eaten everything like at least twice. So my favourite part is Boxing Day sandwich, where everything is like cold cuts and I have like a sandwich this thick. Not with sourdough, was probably with ciabatta this year, but like meats, potatoes, sprouts, by the way, chopped up, just put in there, just have it, and then just that in a sandwich.
Okay, well, that's the end of all my questions, and I think that wraps up the podcast, Rob.
Yeah, it certainly does. Yan, thank you so much for your time. It's incredibly appreciated. And if any spare seats come up at your Christmas dinner, just let us know. And me and Jordan will be around there in a flash.
Welcome to another LabLife, and today we're joined by a very special guest. We're with Reverend Christopher J Lee, who is a retired senior biomedical scientist in haematology and an ordained Anglican priest in the Church of Wales. His career at the University Hospital Wales spanned 44 years, and he also spent some time working in Africa, helping to train laboratory staff, where he developed extensive expertise in parasitology, including diseases like malaria. Welcome to LabLife, Chris. Thanks for joining us today.
Thank you so much, Jordan. Lovely to be with you.
So, could you just start by introducing yourself and, well, how you started to do this career as both a religious priest and a biomedical scientist?
Well, yes, of course. My name is Christopher Lee, which I share with a film star who was famous in the 1960s and 70s for playing horror movies about vampires and Dracula. So it was always a great icebreaker when I said my name was Christopher Lee. And it was curious because by coincidence, the speciality that I chose eventually to study was haematology. And so there were many comments about, you're having a laugh, somebody from haematology or blood bank by the name of Christopher Lee. But there it is. And I started actually in 1971. But in the first couple of years of my career, I studied the main disciplines of pathology. And in those days, you had to choose which you wanted to do. So I chose haematology, which fascinated me from the beginning. And it happened. I guess I had an attraction for the public service because my father was an electrician. And he worked at the Cardiff Royal Infirmary, also for 40-odd years. And things used to break down with the electrics and so on and so forth. And I remember as a kid, he used to take me in when he was called in to fix something, and that. So I used to see these people walking around laboratories in white coats and things like that. I never liked sitting in exams very much, mind.
So, Chris, were you always associated with the church and had a higher role within the church at the same time?
Well, it's true to say, I was a kind of a dyed-in-the-wool Christian, if you like. I was just a regular member of the congregation for many, many decades. But I eventually felt the call to ministry whilst I was working. And so I embarked on another degree course at Cardiff University doing what was called practical theology.
Do you think working at the same time in the church and having a senior role there actually conferred any advantages to your role in the lab and being a haematologist? Do you think it helped at all? And what were those advantages?
I think it helped no end because it gave me actually both disciplines fed off each other because you need an empathy. Anything in the public service needs an empathy, and the scientific service too. But as a Christian, it would give me an opportunity to show in a practical way what it was, to live with a faith. I always felt that the example that I led and the people that worked either with me and for me were inspired by the love and the gentle nature and the acceptance of the talents of everybody.
And on the topic of empathy, you actually took part in a project, didn't you, in Africa at one point in your career, where you're helping to train lab technicians across the sub-Saharan continent. Can you tell us a bit more about that project and what exactly you were doing over there?
Yes. I think it's probably fair to say, Jordan, it was amongst the very highest points in my life. The project itself spanned quite a number of years. And I had an email popped up in my inbox one day and was invited to the headquarters of the Royal College of Pathologists. And I found myself being elected the head of the Zambian delegation. The project took place in several Central and Eastern African countries, namely Zambia, Zimbabwe, Uganda, Tanzania, and Kenya, concentrated mostly on the hospitals and the hospital laboratories in Zambia, although we did go to the others and held retreats, they were called, but they'd be attended by delegations from all of those countries and various towns and cities within those countries. When everybody was gathered together, which happened on a few occasions, there were probably 300 or 400 people present.
And what kind of work were you doing in the laboratories there? What kind of work were you doing to help train the staff?
Right. Now, the project was called Lab Skills Africa. And the idea was to try and up the ante in the sphere of quality, really. In other words, to make sure that the tests that were being performed were actually being performed to the same high standards, or as near as we could get it, to the tests and the analysis that we would do in this country. Not always easy, of course, because of the nature of the equipment that was present in the African laboratories, which ranged from places that were fairly well equipped, and indeed some places, like, for example, the Aga Khan University Hospital in Nairobi was very well supplied. But the main thrust really was to help them along with some of the technical issues, and B, in terms of management and the way that we could structure our laboratories and the quality control systems that we could put into place. And of course, each hospital from each country would then do a little project on some particular aspect of what it was they were interested in. And there were people ranging from measuring hemoglobin levels in pregnant women to HIV testing and all the gambit of biochemistry that was available too.
So you brought back with you an expertise in parasitology and in diseases such as malaria into the local lab in Wales.
Well, that was fascinating, I have to say, the malaria side of things, because malaria in particular and blood parasites in particular interested me anyway, even in my early days in UK. So I had a reasonably decent knowledge of malaria and blood parasites before I got involved in the project. But we'd have, like often happens in UK, you get the odd cases that turn up, throughout the year. And I felt that I was on the ball to know how to deal with it and to know how to do it. But of course, in the laboratories in the African places, would probably get as many cases in a day of malaria as we would get in a year, and so the kind of frequency with dealing with it, and the staff that were there on hand who had not the greatest of equipment to actually diagnose it, were really helpful, and some wonderful little tricks of the trade that they'd show me too. Ultimately, of course, eventually there were various little lab-based tests, a little bit like the coronavirus test, and those would demonstrate antigens, and they weren't entirely foolproof. But the laboratories did manage to get some of those, the African laboratories, and use those as well as their microscopy. And so working hand in hand with the folks in the laboratories there was a real eye-opener.
It's really interesting that your experiences in Africa was almost like a two-way exchange, wasn't it? Because on the one side, you'd take your expertise from the UK and how we do things here and the quality control measures and implement those in Africa. And then the expertise and experience from testing for diseases like malaria, you'd take to the labs in the UK.
It was.
Yeah.
100%. And when I described it as one of the high points of my career and even my life, I really meant it, because it was a great experience, and it was a great thing to bring back to this country. And to this day, I'm still in touch with many of the folks in the laboratories.
It's December, it's our Christmas edition of IBMS Pod. I mean, have you got any stories or insights to share with our members from your time working over the Christmas period? It's a special time of year, especially in your role as a priest.
Yes. Well, Christmas was always an interesting time. The science went on, the request went on, often come what may. And people would be coming in via trauma or whatever it was. That it happens Christmas any time of the year or whatever. And I do remember for many decades that we put up Christmas decorations in the laboratory, and wonderful. Somebody in the blood bank one year left the Christmas tree up, and they were busy on 12th night or whatever, and they couldn't take it down. And somebody told them, well, it's bad luck. If you've left it up after 12th night, you've got to leave it up all year. This is a wacky idea, seeing a Christmas tree in a blood bank in August.
They kept it up all year, did they?
They kept it up all year, but it was great fun. The one great aspect that we had and that we have with our society today, and the brothers and sisters that we can share with it, is that there was multifactorial, multicultured. And what a wonderful thing that for Christmas time, our brothers and sisters of other faiths would say, without even asking, we'll cover you. We'll do your shifts over Christmas. And then, of course, we'd reciprocate for Eid or whatever it was, or Diwali, which is also great. I love them all.
Well, to finish off, Chris, I know that you just recently had the opportunity to perform the wedding ceremony for your daughter, Hannah, who's also a biomedical scientist in haematology. I mean, how was that? What was that like?
Well, that was just incredible, and it happened by sheer coincidence that the vicar of the church that she was being married in moved on to Birmingham, and he rang me up and said, "Chris, I'm not going to be there. Do you know any vicars could take a wedding?" I said, "It's funny you should say that. I'll do it." And so it was that I did it, and thank God, a great privilege. Mind you, at the beginning of the service, I could hardly speak. So I had to take myself in hand and get a grip, which I did. It all went marvellous. So she's currently working in the haematology department in Southmead Hospital in Bristol.
Would you say you did inspire Hannah to get into biomedical science?
Well, I suppose in the respect that I happened to be in the position that I could arrange to get her to see what it was all about. And so she did.
Well, I wish you a very Merry Christmas, and thank you very much for coming on today.
And the same to you, Jordan. It was such a joy. My work in the community and my work in the church is such an honour and such a joy. And if there's anything I can ever offer to the scientific community in the hospital or whatever, I'd be very happy to do so. It was happy days, and I've been very privileged.
Well, thank you very much, Chris.
Thank you, Jordan. God bless you. All the best.
Thank you.
Thanks so much for listening. We hope you enjoyed the episode. These podcasts are released monthly at the same time the magazine comes out, so whenever a new issue lands on your doormat, head back online to listen to a new episode. And don't forget that these podcasts can be used for your CPD. Take care and bye.